Once they're separated, now the enzyme that reads this in order to make a messenger RNA molecule, now that enzyme can bind. And so here's one difference between DNA replication and transcription, right? That difference is the type of enzyme that does it. So if you remember, when DNA was being replicated, when DNA is used to make another DNA molecule, the enzyme that does that is called DNA polymerase. Well, here during transcription, what the cell wants to do is not make DNA, but to make RNA instead. And so the enzyme that's used to make RNA is called RNA polymerase. So the thing to keep in mind here, it's kind of easy to confuse what the different polymerases do. But if you just remember that this word in front of polymerase, that tells you exactly what this enzyme is going to make. So RNA polymerase makes RNA. DNA polymerase makes DNA. Right? So again, here we're talking about transcription, which is using a DNA molecule to make RNA. Because RNA is being made, you know the enzyme that does that is called RNA polymerase. It turns out that there is a particular sequence. So how about if we define this? RNA polymerase, it's the enzyme that makes RNA. It turns out that there's a particular sequence on the DNA molecule on which the RNA polymerase enzyme first binds to. Right? So in other words, RNA polymerase has to know where to attach to the DNA in order to begin transcription. There's a sequence on the DNA that kind of directs the RNA polymerase where to bind. That sequence on the DNA where RNA polymerase binds to is called the promoter region. Right? So the promoter region, again, it's the sequence on DNA where RNA polymerase first binds. Another way of thinking of the promoter region is that it's the region on the DNA that tells the RNA polymerase where the start of a gene is. Right? Because an RNA polymerase, it's going to transcribe a gene. It has to know where the beginning of that gene is, though. And the promoter region kind of signals that. It signals to it where is the start of the gene. In other words, it tells the RNA polymerase where to bind, right, to the DNA molecule. So after RNA polymerase has found the promoter region on a DNA molecule, and after it's now bound to it, now it can start reading this information on the DNA in order to make RNA out of it. So basically it goes down the DNA molecule and it helps the complementary RNA nucleotides attach. So if you remember, what's complementary to a G is a C, so it helps that attach. C's complementary to G. Whenever it sees an A, remember now that since RNA polymerase, it's building an RNA molecule, RNA molecules have U's instead of T's. So whenever there is an adenine, RNA polymerase will add a uracil instead of a thymine, right? Because it's making RNA instead of DNA. And so it just does that for the entire gene here. So as it goes down, it'll just add the complementary RNA nucleotide to the template DNA strand. It does this until it reaches the end of the gene. And when it reaches the end of the gene, it knows to come off then the DNA molecule. It knows that its job is completed. It turns out that there's a particular sequence, there's a nucleic acid sequence at the end of the gene that kind of prompts the RNA polymerase to fall off. And so if the promoter region tells the RNA polymerase where the start of a gene is, in other words, if the promoter tells the RNA polymerase where to begin transcription, the sequence at the end of the gene that signals RNA polymerase that this is the end of transcription, that's called the terminator region. Right? So in words, what the terminator does is that it's found at the end of the gene, and it tells the RNA polymerase then where to fall off. Right? It tells the RNA polymerase your job's done. You've already transcribed this particular gene. And so bringing this back to the big picture, remember the whole purpose of doing transcription was to read DNA in order to make messenger RNA out of it. And so now here is the messenger RNA. Once RNA polymerase has finished making it, what happens is that the RNA, that messenger RNA, then disengages. Right? It's made, so now its job is to go to the ribosome to tell the cell how to make a protein. you see this DNA here. So DNA is pretty important. You wouldn't want it to get trashed or thrown away. So that's what happens. It's not trashed. So actually, after transcription, the two strands of DNA come back together, and it can be used again, right, to make more messenger RNA molecules. So again, the cell wouldn't want to trash its DNA. It can be used over and over again. Again, big picture stuff. The reason for making this messenger RNA during transcription is that it's the molecule that can now directly go to the ribosome in order to instruct the cell how to make a particular type of protein. And proteins are just a bunch of amino acids strung together. Okay. So here's what we'll do next. So this is basically a general idea of what happens during transcription and what all elements are required during transcription. It requires RNA polymerase, it requires a promoter, and it requires a terminator region. What we'll do next is we'll focus in just on this promoter region here and show you in a little bit more detail what all happens at the promoter region. Right? So more details of what's happening here, and we'll show you that now. So again, if this is say that this is a DNA molecule in a eukaryotic cell, here is one of the genes on that DNA that tells the cell how to make a particular type of protein. Again, we're going to focus in on this area right in front of the gene, the promoter region. And so to kind of simplify things and to kind of really show what's happening, how about if we get rid of this double strand and show you just one strand for now, just to simplify things. Again, at the front of the gene, that's where you would find the promoter region. And so we'll color code the promoter, make it green. Green for go, because again, this is where this is the sequence that tells the RNA polymerase where to first bind and then go to transcribe this gene. So again, we showed you before that what happens at this promoter is this is where the RNA polymerase first binds to in order to begin transcription. It turns out though that RNA polymerase needs a lot of help in order to do transcription. So we'll see that not only does the RNA polymerase bind here, but a whole bunch of helper proteins also bind here in order to make sure that the RNA polymerase does its job. So RNA polymerase, it's kind of like middle school girls, right? Because you never see just one of them go into the bathroom, it's a whole bunch of them. So the same thing here, it's not just one protein, RNA polymerase, that's going to do the transcribing. Instead, it's RNA polymerase plus a whole bunch of other proteins that help it do its job. So here's what we're talking about. If you consider the promoter region on a DNA molecule, you can actually divide this into smaller subregions. There's one region called the Tata box, and it's called the Tata box because there's a bunch of thymines and adenines that make up this particular region. So there's a bunch of T's and A's in this particular sequence. It's at this, or how about if we mention this, there's another region within the promoter that's called the initiation site.